Welcome to another bite-sized episode of Cream Rises Up. According to International Data Corporation, the global data storage demand will grow to a staggering amount of 175 trillion gigabytes by 2025. The storage devices we use today cannot handle such a massive amount of data. Reliability is also of a great concern when it comes to conventional means of data archive. This of course necessitated looking outside of the box and finding that next big thing in data storage and data archiving. After Richard Feynman's 1959 discovery related to the possibility of generating artificial objects similar in functionality to what we find in nature, there were several attempts to manufacture products resembling biological ones. Among these research findings that were inspired by Feynman's papers include the utilization of DNA for data storage. Using a synthetic DNA for archiving data is being touted by many as the solution for the imminent data storage crisis. Researchers are fascinated by the potential of DNA functioning as a long-term data storage mechanism. This is mainly because DNA is incredibly dense and chemically stable. The data quantity of a DNA molecule is astounding. Just 1 gram of a DNA molecule can store up to 215 million gigabytes of data. Moreover, if a synthetic DNA data storage device is placed in properly cooled area, it will stay unspoiled for hundreds of thousands of years with a perfectly intact data. A magnetic hard drive, for instance, stores data in a binary form. Centered on a spindle, the platter rotates and an electronic current reads and writes data onto the surface. Similarly, DNA-based storage requires an encoding and decoding scheme. In this case, scientists chemically create synthetic DNA with certain properties based on the 4 nucleotides bases, which are adenine, cytosine, guanine, and thymine. DNA data storage passes through 2 main processes. These are writing the code by synthesizing it and reading the code by sequencing it. Each of the 4 base DNA molecules is used to hold 2 bits of information. These DNA molecules are synthesized letter by letter with enzyme catalyst and indexed. After the bits are written, they are stored in a container where light and temperature are carefully regulated. Just like all data storage methods we used in the past, DNA also has its own flaws that must be ironed out before it becomes mainstream. The main issues bottlenecking DNA data storage are cost and extremely slow read and write speeds compared to conventional storage devices such as hard drives. There are a number of private firms actively researching how to store data on DNA, the organic chemical that is integral to all known forms of life. A DNA-based storage company called Catalog Technologies is doing a lot of research activities behind the scene to rectify the issues for DNA synthesis and drive down cost and make it viable for mainstream consumption. Catalog's new method for DNA synthesis involves encoding using dozens to hundreds of short pieces of DNA or oligonucleotides. These bits of DNA molecules are then assigned to each bit representing the data. The enzyme merges the oligonucleotides into a single, longer DNA molecule that contains the unique signature of the bit. If, on the other hand, the bit is a 0, the corresponding DNA for its address isn't synthesized. All of the molecules that are produced can then be combined into one single solution and further processed for long-term storage. Reading the data requires sequencing these groups of DNA molecules with the aid of an algorithm which can recognize the unique combination of oligonucleotides present in each molecule. The recognized addresses are then converted to bit of 0s and 1s. Even though this method is significantly inefficient compared to storing 2 bits in every base, it allows the company to create an incredibly compact and stable storage medium. The cost reduction of DNA synthesis is mainly achieved through synthesizing a lot of one specific DNA sequence. By compiling DNA using a small bit of a large volume of pre-made DNA, the cost of synthesis drastically minimized. To make the DNA synthesis, Catalog has built a machine called Shannon, which is based on the inkjet printing machine implementation. Seagate, one of the biggest data storage companies in the world, is also forming a tie-up with Catalog Technologies in order to facilitate the research process concerning DNA synthesis. Seagate's part is thought to involve incorporating a fluid handling hardware that the hard drive company has developed, which could help shrink and automate the process even further. Lab-on-a-Chip, which is Seagate's in-house technology, is going to be a key for Catalog when it comes to producing significantly smaller DNA storage devices compared to the current iteration of Shannon. According to Catalog, when it comes to DNA storage, big companies are not only interested in cold data archival. Rather, these giant multinational corporations are intrigued by the fact that DNA could allow massively parallel operations on the stored data without the need to convert it back to digital form. The company has identified huge parallel database searches and digital signal processing as potential applications for DNA data storage. The case for DNA data storage is mainly emphasized in its sheer power at handling parallel data transaction compared to traditional storage mediums. The computational power for a mere megabyte of data is comparable with processing hundreds of terabytes of data if we employ DNA storage. No doubt that the commercial viability of DNA storage is still some way off. However, Catalog and other big hitters in the industry are slowly making inroads on DNA storage, and it might not be long before we witness DNA-based storage deployed in big data centers and research institutions.